My presentation is a little bit different than anything that you've had before. I'm not a medical doctor, I'm not a researcher, I'm a patient. And I've been a patient for a long time, for 37 years here. And they've worked on me, excuse me, because I had melanoma, thyroid cancer, and prostate issues that they still can't figure out what's going on. But I'm here really on behalf of my fellow patients to share our experiences and observations and suggestions with you who are interested medical researchers. And the presentation is really an accumulation of these experiences and suggestions of a number of my fellow NIH patients. So a lot of this may be oriented to NIH, but I think it's probably holds true for researching outside of the clinical center in the NIH. So why do patients participate in clinical trials? There are really basically two types of patients. Those who are healthy, who want to participate and help others by contributing to medical research and advancing science. And those who are sick, those who are ill, who may have been referred by outside private physicians or who are self-referred, and that's more and more the case these days. In many cases, they've exhausted all other options to take advantage. And they come here to take advantage, and sometimes the experimental and sometimes potentially risky treatments, but that are often inaccessible in any other way. And many are looking forward, really looking for medical miracles. And my thesis is that it's important and beneficial for you as researchers to form a partnership with your patient volunteers. A partnership that's based on trust and understanding, a partnership of peers, one that ultimately advances medicine and also ultimately saves lives. To be successful, your research will in some measure be dependent on the cooperation of your patient volunteers. Their confidence in you will help establish a positive relationship, one that can be extremely helpful to you and for the outcome of your study. At the heart of this partnership for patient and scientists alike is the basic optimism and hope for a positive outcome. So I've created an acronym that hopefully is appropriate as we discuss this, and I call this Q. C is for communication, U understanding, and E for empathy. Communication, verbal and non-verbal skills. You know, you only have one chance to make an initial impression, and it's my position that the principal investigator should be present and take the time for you and the investigator to be thorough in a very private setting. You must explain the role of fellows or investigators, as the case may be, and that you not push for an initial decision from the patient to make that decision to continue to proceed with the project. You need to have a full explanation of every aspect of how the process will work and understand that patients may be intimidated and overwhelmed, especially in their initial meeting, and do encourage note-taking so that they can have a pretty good recall of what was discussed at the meeting. Explain the risk, possible lifestyle effect changes while undergoing treatment. Will they be able to work? Will they take care of their family? What will be the duration of the trial, the frequency of required visits? What are the transportation issues and possible costs, if any? And should they bring a caregiver? Make sure that this conversation doesn't take place in hallways or in the midst of a busy clinic, and by all means, respect their privacy. Sitting when you're talking to your patient conveys a sense of importance and a conversation of peers. And make eye contact. It conveys a sense of direct engagement and that they have your complete attention and focus. And listen intently. No writing, no typing. Today, in this world, it seems that so many clinicians, when they're meeting with a patient, are on their desktop typing away, or laptop, typing away and hardly making eye contact with the patient. I have to tell you, from personal experience, that is extremely off-putting. So listen intently and type after the visit. And review your patient's file and history before, not during the meeting. Think through in advance the likely questions or those that should be asked and cover them, even if the patient doesn't ask. Understand that the experience can be intimidating, overwhelming, and give them time to absorb and encourage note-taking. Be totally accessible between clinic visits. You can be assured that particularly in the first visit, but perhaps on ensuing visits as well, the patient is not going to really be able to absorb everything that you say. And there are going to be questions that arise after she or he leaves you. So be totally accessible between clinic visits. Return phone calls and emails in a timely fashion. Being able to stay in contact with the researcher is extremely important. And if you respond quickly, it does show a sense of your regard for this person and your interest in their well-being. And if the situation warrants it, keep outside primary care physicians regularly apprised of developments. Understanding. Listening is more than a 9-letter word. And you need to assess the patient volunteer's understanding. Do they really get it? Be careful of the overuse of medical jargon, translate technical terms, and understand that many returning patients have been coming for years. Their knowledge and understanding of their illness and research may possibly be greater than yours as a new fellow or researcher. It's an indication of respect for you to recognize and acknowledge this. Respect and encourage patient volunteers' own research. I'm sure you're aware that everybody is on the internet these days, and more than likely those patients who have that degree of interest and availability of computers will be on their Google site and looking for as much information as they can. And some of this research is not going to be accurate, but respectfully correct their misinformation and direct them to reliable sources. Be honestly optimistic. No false hope, but accentuate the positive without eliminating the negative. Understand patients previously treated in the private sector may feel that the clinical research environment is intimidating and a bit off-putting. Be sensitive to that. And make certain that patients understand that they can discontinue participation in the trial at any time without any negative consequences to subsequent treatment. Please never use the fact that patients are receiving free care. This can be offensive to patients, so rather enforce the idea that they are a very important component to the overall process of clinical research, and this process could not be done without them. Remember that the so-called free care is often experimental and sometimes comes with potential risk for the patient. Empathy. The act of simply coming to most large medical facilities and the entire initial experience and even subsequent visits can be intimidating and overwhelming. Ill patients can be exercised, emotional. Remember, for some, you will represent their last hope. Some patients have complained that they're being treated like guinea pigs or lab rats, and feel that their only value is to provide data and resent being made to feel this way. So go out of your way to disabuse your patients of this feeling and make them understand and believe that you truly care about their welfare. Time management. Don't act rushed. The worst thing that can set a patient off is for a clinician to come into the examining room and say, well, I only have a few minutes to talk to you, so let's get on with this. I mean, what could be more important at that particular moment in time than to go face-to-face with your patient? Be on time. Long clinic waits are really disrespectful and off-putting. And to sick and anxious patients, it's extremely off-putting. I can give testimony that having sat in some clinics and waited for an extraordinarily long time for, excuse me, my fellow to come join me. So please put that in your book and do make it an extreme effort. The difficult conversation. Delivering bad news. Every patient is a very real and unique person with genuine fears and hopes and not just an experiment in your lab. A negative diagnosis can include the perception that life as one has lived it is gone forever and there may be little hope for the future. It may require a stretch, but I suggest that you try to stretch and to put yourself in the patient's shoes. It's critical for you to be understanding at this potentially very, very sensitive moment. Perhaps the reason that the trial has to be canceled is because the treatment was not effective, or because the severe and debilitating side effects were overwhelming, or the test that the patient is taking he is not or she is not complying. Whatever the reason, cancellation of a trial can be devastating. So find a private setting. Find someplace really out of the way, quiet, where a very confidential and sensitive conversation can be had. Make sure that the proper staff is there. In all cases, if at all possible, the principal investigator should be there, and anyone that you may feel is important to the patient to be present and present the case to him or her. Allow necessary time. It's awfully important at this moment, particularly at this moment, that you give all the time necessary to give the patient what she or he needs to hear in the most positive, if possible, fashion possible. And when bad news has to be given, do advise the patient in advance to bring somebody with them. Perhaps a caregiver, perhaps a relative, but to be there with them at this very traumatic moment. Be careful with your choice of language. A complaint that some cancer survivors have voiced is the phrase that some physician and others may have used from time to time. Well, you failed your chemo or another treatment, and because by most reasonable measures, the failure that the treatment is the fact that the treatment and not the patient is at fault. And hearing the phrase uttered can contribute mightily to making bad news even worse. This type of pejorative language should never be used and should be forbidden. So please understand that. It's important also to have in advance prepared what other possible options are. If there are other studies in other facilities around the country, make sure that they know about them. If there are other drugs, if there are any other possibilities, this is the time to talk to the patient and make sure that she or he understands that there are other options. Make the patient aware that there is supportive care. For example, psychologists, social workers, or spiritual professionals. All this needs to be arranged in advance so that you're prepared for whatever eventuality, whatever reaction this patient may have. And when it's appropriate, make sure that the physician at home understands what the situation is so that she or he can be prepared to deal with the patient when they return to their homes. Basically, what I've said here is that we take my cue and that make communication, understanding, and empathy cardinal elements of your partnership relationship with your patient volunteer. And never forget that in addition to being your patient, and perhaps just one element in your research, that we patients are human, very real, with sensitivities, concerns, and feelings. When we were presenting this, and I present this to the new fellows here every year, and we have patients who gather here twice a year to be part of a group called the Patient Advisory Group. Dr. Gallen has asked we patients come together and discuss with him what issues we see as we go through the clinical trials here. And I asked at one of the meetings if any of my fellow patients had any comments that they wanted me to convey to the fellows. And I think it's appropriate for me to read to you the letter that she wrote to me and asked to be presented to our fellows. And it goes like this. Dear fellows, we are honored to be under your care. We see you as incredibly curious people, at some advantage to be named a fellow at the NIH. And we want you to know that we appreciate your intellectual fortitude and lack of selfishness. But we also want you to understand that you are not our first shopping experience. We have been poked and prodded and tested by others, your colleagues, your elders, your nurses. You, however, are new to us. We want you to know that we are not our disease. Yes, we understand that you love investigating the disease, and have an urgent desire for investigation. But we are more than our disease. We are grandmothers, worried parents, frightened children. We have hope, love, and compassion. We suffer. We worry a lot. We allow you to be intimate with our inner workings, and we do it over and over and over again. For those of us who come to the NIH time and time again, if we're lucky, we get to repeat our story over and over again. So when you are able, make the time to read up on us. Be informed. We know you like to be informed. You touch the petri dish of our lives. So why not read about us first? A physician once wisely told me, Karen, diagnoses is 90% history, and I've never forgotten that. We're the sum of all of our parts, and your recognition of our wholeness matters in the healing process, as well as the suffering process. So carry on with hope, love, and compassion. Bringing those qualities to the exam room will enlighten and enrich your experience too. She signs the letter, Karen, sister. She's an RN, by the way, MS, sister, mother, grandmother, friend, nurse, patient, survivor. To me, it's the most meaningful statement I could leave you with to understand how we feel as patients going under your care and participating with you in your trials. So thank you very much for listening, and I wish you well and thank you for your interest in becoming investigators. Are there any questions? Well, thank you again.
Good evening. My name is Kate Stoney. I'm a program director at the National Heart, Lung, and Blood Institute. And at the NHLBI, I oversee a diverse portfolio of clinical trials that really span the entire spectrum of the research spectrum, the translational spectrum. And I've been asked to come and talk to you a little bit today about what you need to think about when selecting participants to engage in your research studies, and I'm really delighted to do that. Let me just start by, I think Jerry has left, but I wanted to thank him and really endorse the comments that he made. I think they're really heartfelt comments. I think they're something that all of us as researchers really need to hear and live as we go forth with our research programs. And in that vein, you might notice that in my slides, I really don't refer to patients, but rather participants, because I really like this notion that the people that participate in our research studies are partners, right? They're partners with us as researchers, and identifying them as participants underscores that partnership. Sometimes I might slip and call them patients, but I really mean to call them participants, because that is how I see them. Okay, now I'll figure out how to do this. Okay. So it's an interesting topic to think about, because when you are undertaking a research program, a study that you want to conduct, and you're thinking about research participants and who you want to select, you might think right off the bat, this is not a hard thing to do, right? So let's say you're interested in diabetes, so your participants that you're going to enroll in your study are going to be those who have diabetes, and that seems to be obvious. But I'd like to kind of delve a little bit deeper to get to some of the more intricate aspects of participant selection, to talk about some of the characteristics, to really figure out what are the things that we need to think about carefully when we're thinking about who we want to participate in our studies. So some of the things that you might have questions about, if you think a little bit more deeply, is not only how you decide who to participate in your study, who you want participating in your study, but you need to think about sort of how generalizable the effects that you find in your study, how generalizable do you want them to be to a broader population? And the answer to that question is not a single answer. It really depends on a lot of factors, and we'll talk about those. How do you know that you're studying who you think you want to study? We'll talk about some of those issues. And really importantly, how do you match the characteristics of the participants that you enroll in your studies to the outcomes that you really want to measure? This is a critical factor that I really want to underscore as we walk through this study. And let me just say now that there's an additional lecture that deals with participant characteristics in terms of inclusion of underrepresented minorities and women in studies. And it's archived on the IPPCR archives. It's by Dr. Miriam Kelty, and I very, very strongly encourage you to listen to that lecture. It's very good. She's an expert, extremely well-versed in not only the regulations, but in the ethical aspects. So I'm not going to touch on those, because those kinds of issues are dealt with in her lecture. But I do want to touch on these other issues. So we're going to start off talking about the reasons that you even want to think about characteristics of your participants and how to select those characteristics. I want to spend a little bit of time talking about the translational spectrum of clinical trials. And you know some of this already, just by virtue of the fact that you're here. But I want to talk about that translational spectrum specifically with regard to how it impacts your participant selection criteria. We're going to touch on internal versus external validity. And you will hear this again in this course, and you'll hear it specifically when you talk about significance testing, when you talk about analytic strategies and other design issues. I'm not going to talk about the analytic part here. I really want to talk about conceptually what we mean by internal and external validity and how that impacts our decisions regarding who we enroll in our studies. I want to talk through some of the factors to think about, the characteristics to think about, and how to think about those in relationship to your trials. I'm going to give you an example from a really interesting study that's going to illustrate many of the factors that we're looking at. So let's get started. So some of the reasons to think about participant selection. One is that it's going to, if you think about it carefully, it will help you clarify the question that you want to ask and the study design that's most appropriate for that question. And the way this will happen is by understanding where your science, the question that you're addressing, sits on what's called this research continuum, or you might refer to it as a translational spectrum. But this is the course of science, how clinical trials move. Not a single trial, but the science behind a single question in terms of the trials that are run. So for example, we start in developing any kind of trials, we start with phase 1 trials. And phase 1 trials are really early stage research. They're dealing with safety issues. They're dealing with dose ranging questions. And these phase 1 trials are very, very small, right? Because they're dealing with issues that you don't need a lot of participants to deal with. You really want to know what the safety characteristics are of your intervention. And so they're very small trials, and they oftentimes are healthy individuals. So if you're at that phase, if you're just starting to develop an intervention, you don't even know if it's really safe, you don't really know what the dose is, how much of the intervention you need to deliver in order to have the outcome that you're looking for, if you're in that phase, then the characteristics of your patient population are going to be specific to that and very different than they would be in, say, a phase 2 trial. A phase 2 trial or efficacy trials. These are trials where you're really asking the question under very highly controlled conditions. Does my intervention that I have developed, does my intervention do anything? Is there a signal anywhere? And let me sort of specify what I mean by highly controlled situations. What I mean by that is that you have real clarity, and as an experimenter, as a researcher, you're imposing a lot of clarity and control on the characteristics of your patient, of your participant population, of the environment in which your intervention is being delivered, on the people who deliver the intervention. You're reining all that control in, because what you want to know is, under these very tightly controlled situations, is there a signal? Is there some indication that your intervention is doing something? So in those situations, the patient characteristics are going to be very well described, and your patients aren't going to look, there's not going to be a lot of variability in their characteristics, because you want to decrease that variability so that you can just tell whether there's a signal. Does that make sense? Yes? No? Yes? Okay. As you move forward, let's say you got a signal, and under a phase 2 efficacy trial, you have some indication that your intervention is efficacious. You then might move forward to a phase 3 trial, which is an effectiveness trial. And effectiveness trials are those where you take what works under really highly controlled situations and you test it in a broader context. So you might, instead of testing it in your academic medical center, in the CRC, you might go beyond that and test it in some community health centers. Maybe you'll test it in a whole bunch of PCP offices. It could be in any number of situations, but you're broadening it to see if what works in highly controlled settings works in more real-world settings. And of course, that's where we want to take our interventions, right? We want them to work in the real world, where we really don't, as researchers, we really don't have control. We don't know who's going to be delivering the intervention. We don't know the conditions under which those interventions are being delivered. And we know that patients are going to be extremely variable in terms of their individual characteristics. So phase 3 trials are more likely to mimic those real-world situations. So by necessity, if you're doing a phase 3 trial, then you're going to loosen the control that you have, so you're going to loosen up the patient characteristics. That is, you're more likely to take more individuals that vary with regard to a whole number of factors. And then there are phase 4 studies as well that are even broader, and these are community-based studies where you actually go out into the community, and as a researcher, you have very little control. You may hear these phases referred to as, instead of phase 1, phase 2, as T1, T2, translation 1, translation 2. There are some small differences between phase 1 and phase 2, translation 1, translation 2, but they really do parallel each other. So if you hear translation 2, that means something similar to an efficacy trial. Okay, so what I'm hoping is that in understanding this sort of research continuum, you get the message that the kind of characteristics of your patient population and how clearly you outline what those characteristics are is going to be dependent in part on where your particular science fits on this translational spectrum. And in order to know that, you have to really know the science. You have to know the literature. You have to understand what's been done before you. And you have to understand where you want to be going. Okay. So other reasons to think about participant selection is that the characteristics of your study participants are going to determine how generalizable your findings are. Now, we kind of got a peek at that in the last slide when we talked about the continuum and the fact that we're moving more and more to a more generalizability as you move through the translational spectrum. But let me offer another way of thinking about this. And that way is to think about internal versus external validity. And again, I want to think about this and talk about this in very conceptual terms. There are some analytic analogs to this, and you'll talk about those, and you'll figure out when you get to that point in the series here, you'll understand more how you make that analytic balance. But I want to really think of it at a higher level right now and think of it in terms of conceptual understanding. So internal and external validity are a balance. If you have high internal validity, you have lower external validity, and vice versa. So it's a balance, and you have to choose how much of each you're willing to live with for the particular study that you are carrying out. Okay? All right, so here's a little bit more what they are. So external validity really refers to the extent to which you can generalize your findings beyond the situation that the research study that you just conducted. So if you have high external validity, that means that you can more easily generalize across participants, across situations, across providers, across time, a place. It's more generalizable globally. Okay. So you might think, wow, that sounds good. Why don't we always just have really high external validity? Because that sounds like what we're really aiming for in the end, right? We want to be able to generalize across a large group of individuals and situations. The problem with that is that there's always a risk. There's always a risk for either high external or high internal validity. And if you have high external validity, the risk is that you're going to make an error in your analysis. And the error that you're going to make with high external validity is that you're going to think you see an effect that's not really there. You're going to have a false positive. So the higher your external validity, the higher the likelihood is that you'll make that error. Okay? Analytically, you'll understand this later, but right now, conceptually, I want you to understand it, okay? So on the other end of the spectrum, on the balance, is internal validity. And we talk about internal validity, we're talking about being able to maximize the amount of control that you have. And this is control over all kinds of things, control over your participants, control over how intervention is delivered, control under the situation under which an intervention is delivered. It enables you to control many sources of bias, many experimenter and measurement effects. So this is that high control end of things. Well, that sounds pretty good too, but when you have high internal validity, the risk is that you're going to make the error of a different sort. With high internal validity, the risk is that you're more likely to make a false negative error, that is, not see an effect that really is there. So you can see how you need to balance high external with high internal validity, and that balance is going to look different depending on the question that you're asking and where that question sits on that translational spectrum. So you might guess that early in that translational phase, you might want higher internal validity, right? That's where you want a lot of control. And you're willing to live with false negatives. You're willing to not see an effect that's really there as long as you have a lot of control. As you move along the translational spectrum, though, the balance tips in the other way, and you're increasingly more willing to increase your external validity so that you can generalize across participants, across situations, across time and providers. And there you're more willing to live with the false positives. Okay? So you make that decision based on your individual question that you're asking. And so you have to think really carefully about, where is my question sitting? What do we know? What's most important to me? And when you make that determination, you're really trying to strike the right balance for your particular question. And I like to think about this in terms of, what do I worry about the most? Do I worry about false negatives or false positives? For the question that I'm asking, for the patients, participants that I'm working with, what do I worry about the most? And that will help guide you about which kind, not only where you are on the translational spectrum, but which kind of validity you're more willing to live with. The type of validity that you're more willing to live with is going to help determine how tightly controlled you want to not only have all aspects of your study, but how tightly controlled you want your participant selection factors to be. Okay? Okay. Feasibility. The participant selection factors are going to impact your feasibility. Now, you want to select participants and make the determination about what factors are important based on science. But the reality is that you can have a beautiful, scientific, award-winning question. And if it's not feasible, if you can't implement it, then it's not helpful. So you do need to think about feasibility when you think about these kinds of issues. And feasibility really means a lot of different things. First of all, do you have access to the participants with the kind of criteria that you're looking for, whether they're demographic or clinical characteristics? Do you have access to that population? In addition, you have to think about the participants themselves, and based on the selection criteria that you've determined, are those participants really going to be able, and are they going to be willing to participate in your study? Are they going to be able and willing to adhere to your treatment protocol? Those questions are going to be determined in many respects by the characteristics that you set out. Right? So if you want to undertake a study that requires lots of active participation by your participants, they have to come to the CRC weekly in order to get some kind of testing done, and you are targeting participants who are physically disabled, that is going to be a strategy that may not work for you because it's a feasibility issue. You do need to think about ethical questions, of course. In all of these cases, you need to think about ethical questions with regard to whether you can randomize to placebo, what the adverse event rates are in relationship to the characteristics of the patient population that you're targeting. And the thing I really want to underscore here is that when you think about these kinds of considerations, you need to think about them for all of the arms of your study, not just the active intervention arm. You know, all clinical trials have some kind of comparison arm, right? You'll have a placebo or a usual care or maybe, you know, another type of intervention that you're comparing your active intervention with. And when you think about ethical issues, you need to think about not only is it ethical to randomize this my participants to an active intervention arm, but is it ethical for the characteristics of that population to randomize them to a control arm, to a placebo arm, to a usual care arm? Those are really important, and sometimes we forget about that. We focus so much on the intervention group. Timing of the intervention is going to be really critical when you want to actually capture the participants. And what I mean by that is that in some cases we want participants who have just had an event, right? Those are the participants we want to study. And it may be very difficult for you, feasibly difficult, to access the participants if that time to event is very small. So for example, if you are doing a study of individuals who have recently come to an emergency department because of an illicit drug potential overdose, and you want to enroll them into a study that asks the question of what some kind of outcome is within 30 minutes of arriving at an emergency department. Well, that's going to be very difficult. So the timing of your intervention, it might make perfect scientific sense, but it may not be feasible. When you think about who you want to be in your study, you need to think about that in relationship to the outcomes that you want to target for measuring. Now, outcomes are what we love, right? That's what we really are interested in, what those outcomes are. But you have to think about those outcomes in relationship to the participants that you're including. So outcomes can be lots of different things. You can have event outcomes, which we think of as hard outcomes. These are things like mortality. That's a very hard endpoint. Or various types of morbidity. So from the National Heart, Lung, and Blood Institute, I have a lot of examples that are cardiovascular in nature. That's what I need to tell you because that's what I live with. So a mortality outcome or morbidity outcome might be something like a heart attack. Okay, but in a lot of studies, we really don't have the luxury of measuring these kinds of event outcomes because either events don't happen very frequently, or our sample sizes are very small, or it takes a long time for events to occur. So if you want to study children, you don't want to, in most cases, have a mortality endpoint because it takes a long time, in most cases, for children to reach that endpoint. So what we do is that we sometimes we'll use surrogate or biomarker outcomes. And I've put a little star here because the NIH actually has a definition of a surrogate measure. A surrogate measure is a biomarker that is a substitute, and it's an imperfect substitute for a clinical endpoint. So examples of surrogate measures are something like, again, I'm from the NHLBI, something like revascularization for someone who has heart disease if they have a coronary bypass. That's a surrogate measure for underlying cardiovascular disease. Cholesterol is a very common surrogate measure for underlying cardiovascular disease. And there are many others depending on what your particular area is. And these are very common outcomes because they're accessible and they're more readily available to us rather than event-related outcomes. There are also patient-specific outcomes, and these are referred to as pros. And these patient-specific outcomes are really important, and for some trials, they really will be the primary endpoint, the primary outcome that you're interested in. These are things that only the participant, and in this case the patients, can report on, things like pain or other kinds of symptoms, their quality of life. There are really good ways of measuring these outcomes. The NIH has put a lot of effort into establishing valid and reliable measures of PROS, and if you have any questions about that, I'd be happy to answer them for you. But when you have these patient-specific outcomes, you want to think very carefully about the meaning of those outcomes in relationship to the patient characteristics that you're looking at. And then there are composite outcomes, which are groupings of outcomes, but that doesn't have much relevance to this conversation. So those are the types of outcomes that we are interested in. So what does this have to do with patient participant selection factors? Well, one of the questions you might ask is, are the outcomes that you want to include, maybe your primary or maybe your secondary outcome in your trial, are they feasible to measure given the characteristics of the participants that you are targeting? So what I mean by that is, let's say what you really want for an outcome is a hard cardiovascular event, like a myocardial infarction, a heart attack. Let's say that's the measure you're looking for. If you choose to enroll, in this example, 50-year-old essentially healthy women, you're not going to see too many of those outcomes in your 5-year trial. It's just not going to happen if they're essentially healthy women, middle-aged women. So you need to match the characteristics of your participants with the outcomes that you need to look at. And in this situation, you'd either have to change the characteristics or you'd have to change your outcome. You also want to think about whether your outcomes can change within the parameters of your trial, the length of your trial, and the follow-up period that you've designed for your trial, given the patient characteristics. So, for example, again, if you have, let's say you're doing a trial of, let's say you want to look at carotid IMT. That's the amount of blockage in the carotid artery over the course of your trial, and your follow-up period is 6 months, and you're looking at essentially healthy people. That's not going to happen. So you need to either change your outcome or change your participant characteristics. If your focus of your outcome is going to be on patient-reported outcomes, PROS, then you need to think about the characteristics of your participants and whether they will be able to give you those kinds of outcomes. So, for example, the example I have here is if you are looking at pain in children, if you're enrolling 2-year-old children to report on their pain symptoms, you will get an outcome, but it may not be very reliable. Now, I will preface this by saying that we can get good, reliable patient-reported outcomes from children if you use the right measures, but you have to think about that in the context of your trial. The bottom line with all of these things is that decisions that you make regarding the characteristics of the participants that you study are going to influence the causal inferences that you can make. And the bottom line with all of our trials, what we want to do is to be able to make causal inferences. You want to be able to say, the reason I found this outcome is because I employed this intervention. That's a causal inference. And you want to be able to do that. You want that inference to be high. So you want to make these decisions very carefully. So I want to move now. Are there any quick questions? Okay. I want to move now to talking about some of the things to think about in outlining your participant characteristics. And we'll talk about entry criteria, contextual factors, access, adherence, and recruitment and retention. So let's start off with entry criteria. When you are starting a study, before you start a study, you want to establish what your criteria are for entry. And you're going to establish both your inclusionary criteria as well as your exclusionary criteria. And by the way, you also, I have a note here that you want to identify what the target of the intervention is. Most of the time it's going to be an individual patient or participant in your study, but sometimes it could be a dyad or a family, sometimes it can be a provider or a healthcare delivery system. It's important to identify that straight up. So let's talk about entry criteria. Interestingly, the main purpose of inclusionary criteria is different than the main purpose of exclusionary criteria. Okay? So with inclusionary criteria, the main purpose is trying to identify the participants who are going to be most relevant to the outcomes of your study. That's the main purpose. You also need to think about inclusionary criteria because you need to report on it. You need to report in your consort statement what your inclusionary and exclusionary, your both inclusionary and exclusionary criteria, and you need to be able to identify individuals that you've identified that are fitting your inclusionary criteria. And then you need to balance between participant variability. So what do I mean by balancing between participant variance? So your participants are going to have lots of different characteristics. And when you include participants, when you set out what your inclusionary criteria are, what you need to think about is, again, how tightly controlled you want those inclusionary criteria to be or how loose you want them to be. So if you say your inclusionary criteria are anyone between the ages of 18 and 70, that's a wide open criteria. And that's fine, but it's going to have lots and lots of variability in it. For some studies, that's going to be appropriate, but for many studies, it won't be appropriate. The variance will be too wide. So one of the examples I give here is that let's say you're doing a study of migraine. You have a new treatment for migraine. So you have to set out inclusionary criteria. So maybe you'll say, I'm going to identify the type of migraines. I'm going to identify the duration that the migraines in any individual participant occur for, and the frequency of those attacks. And I'm going to specify what other medications participants can use when they're enrolled in the study. Those are inclusionary criteria. Those you report out on, and those are going to target for this question specific characteristics that are relevant. Now contrast that with exclusionary criteria. The main purpose of exclusionary criteria is to establish safety, to control confounding factors, and for feasibility. So when you set out the criteria by which you are not going to allow someone to participate in the study, the most important reason to not allow someone to be in your study is for safety reason. If the study is going to be too high risk for that particular characteristic of a participant, you don't want them in your study. It's going to be too risky. If you're not sure about the safety issue, you may use some of the criteria as exclusionary criteria. There may be things that characteristics that participants bring with them that might confound your findings. Let's say you're trying to test a new drug, and you want to make sure that the effects of that drug are clear. You may not want to allow other medications to be used because they could confound the effects that you're looking at for your intervention. And then feasibility, which we've talked about before. So the example I give here is, let's say you're testing a new medication for high blood pressure, and you may want to exclude people with heart failure for safety reasons. People with heart failure are complex and difficult to treat, and so we may not want to include them in a trial of a new medication. You may exclude people with diabetes. Diabetes and high blood pressure are often comorbid, but there may be some confounding factors. So you may decide to exclude those participants. And feasibility issues, if you have to do lots of testing, you may not want to include individuals who are bedridden, for example, because it wouldn't be feasible for them to come to the clinic frequently for testing. So the reason for excluding participants is very different than the reason for including participants. Now, one of the questions I get all the time is, when do I establish what the inclusionary and exclusionary criteria are? And the answer is very, very early. You want to establish this before you start your study, obviously. You need to establish it before you go to your IRB for approval. You need to establish it before you get funding, because it's going to be so critical to the success of your study. So you do it very, very early on. The reason these criteria are so important is because this is an easy way for unintentional bias to creep into your study design. If you're not clear, and I mean crystal clear, what your inclusionary and exclusionary criteria are, not only for you as a PI, but also for any staff that you have who are going to be recruiting participants. If you don't have crystal clear inclusionary and exclusionary criteria, it's possible that you may end up with a bias sample. Your own bias in wanting to enroll a particular participant into your study may creep in unless you have really clear reasons to include them or exclude them. So I encourage you to do this early, to be very, very specific, to write it all down, and to make sure that everyone on your staff understands what these criteria are. So the next question that I always get is, well, can I change them? And why do people want to change their entry criteria? The most difficult part of doing a clinical trial is what? Recruitment. Everyone knows that. So if you start a trial, you have entry criteria that's clearly laid out. You can't enroll adequately. You're not meeting your milestones. One of the things that people want to do is change their entry criteria. You can do that, but you pay a price. So the price you pay is the price that you may actually allow some of that bias to creep in. So if you're going to go down this road and change your entry criteria, you want to make sure that you do it in as unbiased a way as possible. You don't change your entry criteria when you're sitting across from someone who you really want to enroll in your study. They're really perfect. There's one little inclusionary criteria they don't quite meet, but they're close, and you really need to meet your milestone this month. That's not the time and the way that you change your entry criteria. If you need to change it, you go to your DSMB, you go to your IRB, you do it in as unbiased a way as possible, and you do it extremely infrequently. So if you're going to do it because of recruitment issues, do it once. Don't keep coming back to it. Again, because you do not want to bias your sample. Context. There are lots of contextual factors, and I'm not going to go through all the contextual factors that are potentially factors to think about when you're thinking about who to include in your studies. But context is really important. And just like Jerry was saying earlier, when you are a researcher and you are engaging with a participant in your study, you have a relation. That is a relationship. And that relationship has context. And not only that, participants who come into your study have a whole world of context, almost none of which you're aware of. Some of which you may be aware of, but almost none of which you're aware of. So by context, what I mean are things like personal experiences, background, culture, their social environment, how a participant feels on that particular day, the relationship that you as an experimenter or researcher have. All of these are contextual factors, and they make a difference. And I'm going to prove to you that they make a difference. I've got a couple of studies to show you. So these are the findings from a study called SWAN, which is a study. It's a longitudinal study. It's not a trial. It's a longitudinal study of middle-aged women. And the purpose of SWAN was to look at women longitudinally as they go through, as they age, and to look at a variety of health outcomes. One of the outcomes that they were particularly interested in was metabolic syndrome, incident metabolic syndrome. But they took a lot of measures, as often we all do as researchers. We take a lot of measures. And one of the measures that they happened to take were some measures on early childhood trauma. And the SWAN investigators had some measures of metabolic syndrome at 2 different time points. And when they looked to see how metabolic syndrome was progressing in this large cohort of women, somebody had the very clever idea to look at the progression of metabolic syndrome in relationship to early childhood physical abuse. Now, there is a literature that suggests that that might be something interesting to look at. And what they found was that the percentage of women who actually were more likely to develop metabolic syndrome was much, much higher, twice as high, in women that had experienced early childhood physical abuse. The experience of having early childhood trauma is a contextual factor. And I'm not suggesting that you go out, no matter what your question is, and ask everyone about early childhood trauma, but I am suggesting that you look at the literature carefully in terms of these contextual factors and think carefully about what measures you need to be taking in addition to your primary outcomes, because context is important. And I have another example in case this didn't sway you. And this is a finding that people know about. It's a very familiar finding, but it's interesting to point out. This is a study looking at ambulatory blood pressure monitoring. They were looking at cardiac events. Again, disclaimer, I'm from the National Heart, Lung, and Blood Institute, so that's what I talk about. And what they looked at was they followed up individuals who were hypertensive and taking hypertensive medication. And they followed up to find out what the survival was in these patients. And what they found was that I didn't show you the first curve, which was something like a curve in between here. What they found, though, was that if you separated out patients that took at least one of their blood pressure medications before bed from those who took all of their blood pressure medications in the morning, you find very different survival curves. And as I say, it's better to take it right before you go to bed, by the way. And as I say, this is a familiar finding to us now. We know that the timing of medication is important. And for blood pressure medication, it's important to take at least one of your medications at night. But what I'd suggest to you is that the time at which a participant takes medication is a contextual factor. So again, you wouldn't know that unless you read the literature, and it is a big literature now, an increasing literature, but it's contextual. So context is important. That doesn't mean that you need to measure everything, because you can't, but you need to think about context when you are thinking about your participant characteristics and your outcomes. So access is another feasibility issue, right? Can you access the particular participants with the particular characteristics that you're looking for? And you need to think about this early, because, again, you can have a fantastic study. It could be the best science in the world, but if you can't get participants into your study because they don't live in your geographic area, or they don't come to your clinic, or you in any other way don't have access, then you can't carry out the study. And one way of increasing access to the participants that you would like to study is to build an interdisciplinary team. Now, there are lots of good reasons to build an interdisciplinary team. One of them is that it helps enhance access to participants that you might not have access to, but someone on your team may have access to. And of course, as you build this interdisciplinary team, and as you have access issues, you may need to have many sites in your trial, and that's okay. It has its downsides, but that's okay. But again, thinking about these in the context of the patient characteristics that you are identifying. I'd like to make a plea to not include your own patients or your own practice in your research. Separation helps you maintain equipoise and deliver good clinical care. We'll talk a little bit about recruitment and retention because it's such a critical issue in running a trial. And I actually do a whole couple of lectures on recruitment and retention because it is so important and because it's complex. It doesn't seem that way, and we all start our trials saying, okay, we're just going to recruit people and they're going to stay because they love us, but it's hard. It's really hard work. And so it's good to think about it at all phases of your trial. Most of the trials that don't complete, and a failed trial is a trial that doesn't complete. Most failed trials don't complete because of recruitment issues. Sometimes retention, but mostly because of recruitment. So what do you need to think about in terms of both recruitment and retention? And probably the number one thing to think about is the burden that you, in your protocol, are putting on the participants that you are selecting to be in your study. And that burden is part of the assessments that you do, the measurements that you do, as well as the complexity and the duration of the treatment. So if you're planning a 5-year trial that requires 4 years of follow-up from your participants, that's a heavy burden. If you're requiring participants to come into your clinic every day for a couple of weeks, that's a heavy burden. And the higher that burden, the more difficult it's going to be to get participants into your study, no matter who they are, and to retain them in your study. And it's going to be even more important depending on the characteristics of your participants. Those characteristics are things like how healthy your participants are or how sick they are, what other needs they have. And remember that those needs extend beyond their own personal needs to their family needs as well. So if you want to do a study of, childbearing women, healthy childbearing women, for example, it's great to identify that population for study, but they may have childbearing issues, so you may need to provide childcare for them. Other participants may have other kinds of logistics issues like transportation issues. Everyone has parking issues because it's almost always difficult to park. So those kinds of things are things that you want to think about and try to do something about to reduce the burden. I have here this notion that intention to treat is the analysis that you will do. An intent to treat analysis means that you will analyze everyone that you randomize into your trial. You will all do intent to treat analyses. And that means that as soon as you randomize someone into your trial, they're yours. They are yours. That means that you will carry them throughout the duration of the trial. They may not give you any more data beyond the first visit, but you must include them in your analysis, in your primary analysis. And what that means, you'll talk about this in your analytics section later, but what that means is that you want to be really careful about who you randomize into your study. So I know that a lot of you have had experience in trying to enhance recruitment, and you do a lot of things. We'll talk about what some of those things might be. And what you want is to encourage people to sign up for your trial, right? That's what you try to do when you make these active and really intense recruitment efforts, and that's a good idea. But I would suggest to you that when you are doing your recruitment efforts, what you actually do is make it hard. Make it hard for participants to sign up for your study. And the reason for that, the reason I say that, and I don't mean make it feasible, make it hard in a feasible sense, but I think you want to be really, really brutally honest with participants in terms of what it will take to be in your trial, what they will have to do. I recommend that if you have a group recruitment effort, that is, you hire a room like this, you have a room like this, and you have a whole bunch of people come to listen to your pitch about being in the study, you have discussions about what the downsides of being in your study is. Have the discussions outright, because I can guarantee you that participants are going through those things in their heads, so you might as well get it out on the table. And by doing that, you may get a few fewer people to actually enroll in your study, or say that they're interested in enrolling in your study, but you will get people who are more likely to be retained in your study. And it is more important to get people to be retained in the study than to get a lot of people who are enrolled and then drop out. That hurts your power, it hurts your ability to make causal inferences. It's much more important to retain participants. So as you're doing your recruitment efforts with all of the criteria that you've laid out in terms of who you want to enroll and who you need to exclude, be really critically honest about what it's going to take for participants to be in your trial. So oftentimes people want to know, I've tried everything. What more can I do? So I want to walk through just a few things. We're a little bit, it's related to what we're talking about, but it's important, I think. So what are some of the things that you can do to enhance recruitment? And one of them is to use this layered approach, which is, another way of saying a multi-pronged approach. Do not rely on one approach for your recruitment efforts, because one approach is probably not going to do it for you. So if your IRB will allow it, use social media. It's very effective. You need to go through your IRB, though, to make sure it's allowable. If it's not, you can use community-based kinds of strategies. And even though you wouldn't go through social marketing strategies, you may have community-based outreach programs that you can piggyback onto. The old-fashioned way is to do targeted distributions of mailings. It's old-fashioned, but it works. You get about 1%. And that's okay, because it's pretty cheap. But it is an effective strategy. But you have to have a lot of people available to send out your mailings to in order for it to work for your particular study. A lot of people do presentations at health fairs or community settings. That's a great idea. I encourage you to do it. None of these are going to give you, half of your sample. They're all going to be this layered approach that you take. One thing that I don't see as frequently, but I really recommend it to you, is to pair up with one or two others who are doing research in a different area, but maybe have sort of similar criteria, entry criteria that you do, but different exclusionary criteria. Because what might happen, and I've seen this done very successfully, what can happen is that somebody who is not eligible for your study may be eligible for the study next door, and vice versa. And if you can partner with other researchers, and you must do this with permission of your potential participants, it can be a very effective way, because you have somebody who's already interested in research. They already want to contribute. And so once you have that person, even if they're not eligible for your study, they may be eligible for another study. So think about that referral process. Set your milestone goals reasonably. That is your recruitment milestones. Make sure that you are being realistic. And then in some cases, you can employ a run-in strategy. And this is not universally applicable for all studies. You have to think about whether it is appropriate for your study. But a run-in strategy is when you have potential participants before you randomize them. You consent them, but before you randomize them, you have them do something. And you have them do something similar to what they would do in the study, but it's something that both arms would do, or all 3 arms, however many arms you have. So if you have people that you want to record in a diary every day, send them home for 2 weeks and ask them to record whatever you want them to record in a diary. And that run-in period can help you identify who will be adherent and who will not be adherent. It's a much more self-selected population that you end up with if you go through this strategy. So if you're looking to do something on the effectiveness end of the spectrum, it may not be the right strategy. But if you're looking to do something on the efficacy end of the spectrum, it may work for you. And of course, you always want to employ strategies to make sure that you have a representative sample. Retention is as important, maybe more important than recruitment. If you can get people into your study but you can't keep them there, that's a problem. So how do you improve retention? And there are lots of things you can do. Probably the most important is to think about burden and think about how you can reduce burden. The other thing to think about is, by reducing burden, what I mean is shorten your study duration, shorten the amount of questionnaires people have to fill out, shorten the number of visits they have to do, or make it easier for them to do those things. Provide vouchers, go to their home sometimes, go to a more central location. There are lots of things that you can do depending on your particular study. But make your intervention as simple as you can. Complex interventions are a reason that participants do not adhere and eventually why they drop out. If you can optimize the visits, you know they're coming to the CRC for a medical visit, make sure that's also a study visit as well. That helps. Reducing barriers, we've talked about that. Providing incentives. Incentives don't have to be costly, but they do have to be kind of meaningful. So a refrigerator magnet, pencil, maybe not, but something that really means something to participants. If you're getting participants a medication that, for example, really increases dry mouth, that's a burden for participants. Nobody likes that. So maybe you give them a water bottle that has your logo on it. That's a meaningful incentive because it tells participants that you understand what some of these side effects are and that you're trying to help them out. Make your visits, set up your visits early in the process. If you set each visit at a time when you have a current visit, then so what am I trying to say? Set up the follow-up visits early on and set them all up at the same time so that participants know what they're getting into and they know when they're going to come back. If you do that, they're more likely to be able to make those visits. So adherence. The only thing I really have to say of adherence is you need to measure it, and it's a hard thing to measure. You need to, as much as you can, enhance it. And by adherence, I mean participant adherence, obviously, but also provider adherence. So whoever is delivering your intervention, you want that person to be as adherent as possible as well. And we talked about using a run-in phase in order to increase adherence. And this is an example that many people actually use. If you're doing a study of obstructive sleep apnea, then the treatment of choice for most patients with obstructive sleep apnea is to use CPAP, right? To use continuous positive air pressure for them. But it's very, very burdensome because it's the mask that people have to wear. They have to wear it at night. Nobody likes it. Even if you're not running a study, patients who use it don't like it. But some people will do it, and some people absolutely won't do it. So how do you know who those people are beforehand? Because if nobody is going to wear the CPAP, then you're not going to be able to find out the answer to your question because that's part of your intervention. So in that case, what you might want to do is have a sham run-in where you actually have people wear CPAP. You don't deliver anything, no positive air, but they wear the mask. And people that can't adhere to that likely won't adhere to the intervention. So that's an idea of how you can do these sham run-ins. There are ways to optimize adherence. I recommend that you get someone on your team who knows how to do this. There isn't a science to adherence. One of the strategies that can be used is called motivational interviewing. It can be used with participants and it can be used with providers. And it is a way to increase engagement of your participants in your study and providers. But you can't learn how to do it from a book. You need to be trained in it. So if you want to employ this strategy, if you have a large trial and that's burdensome and want to employ this strategy, it's a good idea to think about finding somebody who can do it. This orientation session is something that I was alluding to before, when you have a group of people and you're kind of telling them about the study to see if they want to participate. This is before you actually enroll anyone, before you randomize anyone. You're kind of laying out your case for why they may want to participate. This is a great opportunity for you to provide information and to develop this relationship with potential participants. I really like, it's not appropriate for all studies, but if it is appropriate for your study, I really think this is a fantastic strategy. It enables you to answer questions. It enables you to get fairly adherent people, because I've already come to an orientation session, people who are interested in the study, and it enables you to give them the case why they shouldn't enroll, all the reasons why they shouldn't enroll. Of course, you're also going to talk about why they should enroll in the study. But it really gives you an opportunity to get people into your study who you may be able to retain into the study. Contact backups, these are family members. If you can get those from participants, that's a good idea if you're doing a longitudinal study. People move and sometimes they forget to tell you. So it's a good idea to have a backup. And again, you get that from the participants with their consent on the consent form. And then if you can maintain contact, again with trials that are longitudinal, if you can maintain contact with participants, one way to do it is to phone them if that's part of your intervention. But you know, it's really nice to send birthday cards. It doesn't cost very much, and people really appreciate it. Again, it's part of this relationship that you're building. And newsletters are also very effective. They take time, but they're very effective. So I want to end with an example of a study that kind of highlights some of the points that I've tried to lay out for you. This is a study called Enriched. It was done a couple of years ago. It's over now, called Enhancing Recovery in Coronary Heart Disease Patients. And the basic premise of Enriched comes from data like these. This is not the Enriched data. These are other data from an earlier study. And it's a phenomenon that we know very well. And the phenomenon is this, that after somebody has a heart attack, if they're depressed, they are much more likely to die than if they're not depressed. So what you can see, this is a months post-MI, post-heart attack. And within 2 months post-MI, as you can see here, we see a divergence of participants who become depressed versus those who do not become depressed. And you can see the mortality rates just go up. It's a phenomenon it's a very real phenomenon. Okay. So the Enriched trial said, we need to do something about this. Let's see what happens if we treat the depression. Let's try that. So that's what the Enriched trial was. And they also knew that depression was very strongly linked to social support. So they actually had an intervention, a dual intervention, to target low social support and high depression. And so the hypothesis was that they wanted to test whether treating depression and low social support right after a heart attack was going to reduce death rates and non-fatal recurrent heart attacks. Simple study, right? It was not simple. So ultimately, they enrolled almost 2,500 heart attack participants that either presented with depression or low social support, or both, very commonly both. And they randomized into a group that got usual care versus one that got a psychosocial intervention to treat the social support and depression. And their endpoints were cardiac mortality and recurrent heart attack. And they followed them for almost 3.5 years, and they were totally masked in terms of their primary outcomes, and they did an intent to treat analysis. So here's what I want you, no, this is not what I want you to see. Their inclusionary criteria were that they were going to enroll people within 28 days of having a heart attack. Now, they wanted to enroll them quicker. They wanted to get them more quickly than that, because you saw in that graph that the divergence happened at 2 months. So they wanted to enroll them as quickly as possible, but they knew that it would not be feasible to enroll them much more quickly than this. Why is that? Well, you have a patient just have a heart attack, they're in a hospital, their family's involved, they have other things to think about besides your study. So they settled on recruiting them within about a month after a heart attack, and they had a bunch of criteria. They had some inclusionary criteria that that could substantiate the fact that they had a heart attack. They looked at enzymatic increases, and then they identified whether they were depressed and/or had low social support, and there are good measures to do that. Here's what I want to show you. This is the concert diagram. They screened almost 34,000 people. 34,000 people to get 2,481. So they screened this number. Some didn't meet criteria for having had a heart attack, right? But not that many. Most of them had a heart attack. So they met that criteria. And then some of them, fair number, 7,000 of them almost, weren't depressed or had low social support. So they didn't meet those criteria. But look at this. 23,000 didn't meet their medical eligibility. Given what we've just talked about, what does that mean? Did they do something wrong with their inclusionary or exclusionary criteria? Yeah. I can't say it's wrong. Some of the best researchers around were doing this study. It's not wrong. But ultimately, when you step back and look at it, this is where they had the problem. This is where it happened. So it wasn't that it was wrong, but they had to screen so many people in order to get 2,500 people because of these eligibility criteria. Scientifically, it was the right thing to do, but it made for a very difficult study. And to operationalize this a little bit more clearly for you, what this means is that for every 100 participants that they screened, these weren't people that called up and said, I want to participate. These are 100 participants that they screened. Only 7 were actually enrolled and randomized into the trial. That's a lot of work. Another way of thinking about it, in order to get 1, they had to screen 14. And of course, they had a lot of sites, and not all the sites were really able to adequately enroll participants. And that's something that when you have a multi-site trial, you will find that sites recruit at different rates. There may be reasons to keep sites that have low recruitment because they're recruiting particular characteristics of the patient population that you're interested in. But it is a phenomenon that occurs fairly frequently. So, there's been a lot of talk among the investigators about why there are these differences, why there are these problems with recruitment and retention. And some of them had to do with access. Some of them had to do with the fact that they were recruiting at very research-heavy sites. So there was a lot of research going on. And you have to think about that. Is there a lot of stuff going on in this area where you're going to be recruiting? They didn't have adequate collaborative teams, and so that reduced their ability to recruit. And their burden was high. So these participants had to come in and participate in a psychosocial intervention, which always is a little bit more burdensome than taking a pharmacologic pill. And they had a lot of assessments, and the study went on for 3.5 years. Right? So this was a very burdensome study. But the real key is here. They had really restrictive eligibility criteria, as you can see from the exclusion, the high number of patients that were excluded. So let me walk through a couple of overall conclusions. Know your literature. Know the history of your literature. Know where things sit. Be able to identify contextual factors. Know where your question lies on that research continuum. That's really critical. Make sure you're designing the right study for the right time, for that literature. Make sure you have clarity on your question. And this sounds kind of silly, like, of course I have clarity on my questions. I want to know if this does this. But understanding the complexity of your question really means that you have to dive deep into some of the issues that we're talking about, and more as well, to understand what your all of the complexities of your question. Have a really good, very specific, targeted understanding of the patient characteristics that you want to target. Make sure the participants match the outcomes, your primary outcomes. Make sure it's a good match, it's a feasible match. And make sure that you're asking the right question at the right time. Going back to internal versus external validity, figure out in the context of your question what's most important. Or in other words, thinking about what you worry about the most. False negatives or false positives. And that's all I have. Are there any questions? If, as you're reading through your material, if you have questions, feel free to submit them to me. I'd be happy to answer them for you. Thank you.